So we're going to pick back up and talk about evolutionary development, this will move us into our next section, our next topic, which is development. And so this is kind of the precursor to that, and that's talking about sort of the mammalian brain and realizing that the brains of mammals are very complex and that a lot of our behaviors in animals and in humans, which humans are animals, is not really one particular single part of the brain, and usually involves many different brain areas and levels of processing. And so we're going to kind of move through the simpler part of the brain to where we have more complex activity. So if we look at the evolution of sort of how the brain has come about, if we look at a fish, amphibian, or reptile adult brain, you're going to see the front, middle, and hind brain very similar to what you would see in a mammalian brain. And so you have the prosencephalon, which is the front brain. This is really important for olfaction, which is the sense of smell. Mesencephalon, which is the middle brain, which is important for vision and hearing. And rhomboencephalon, which is the hind brain, important for movement and balance. And so these are what is considered sort of the older parts of the brain. Think of any animal needing to be able to smell, hear, see, needing to be able to balance, be able to move. And we know that, for example, the spinal cord is a part of that hind brain. And this is really good to know. This idea is how these portions developed out into what we see as big structures in a human brain, because it can kind of help us understand where certain structures are and why they are important to survival. If we look at mammalian brains, we still have, of course, the prosencephalon and the hindbrain, but we see more structures and segments that come from these main sections. So, for example, the prosencephalon, we see having the diencephalon and the telencephalon. So the diencephalon is what referred to as sort of the between brain, and that's a series of subcortical structures. And then we have that telencephalon or end brain, which includes the cerebral hemispheres as well as the cortex. Now, when it comes to the hindbrain, that's split into two major parts in the mammalian brain, which is metencephalon, which is referred to as the cross brain, which includes the cerebellum, and the myelencephalon, which is the spinal brain, including the spinal cord. And if we look at these different structures and how they are segmented out, this is going to help us later when we're into the human developing brain, because you can kind of see how these structures have sort of formed, and then over the period of from conception, looking at brain development, how it develops in utero, can show you sort of where the brain ends up forming. And there's a really nice video that we have in the development slides that'll get us through that. So these are all color-coded, so you can kind of see we have the forebrain, we have the midbrain, and then we have the hindbrain in a vertebrate embryo. And then here we have the spinal cord. And then if we look at a mammalian embryo, you can see that it's the same major idea in terms of structures, but now this area here, this forebrain area, is split into the telencephalon and diencephalon here. And then here, if you look in a human brain, that becomes the neocortex. That becomes the basal ganglia, the limbic system. That's important for olfactory. That becomes those lateral ventricles. And this is what makes up this major forebrain area. Now, notice that this is color-coded, this really big blue area here, right? And including right here. Not here. Not the cerebellum yet. And then if you look here, the mesencephalon, which is the midbrain here, okay, it's still, you can see it in the mammalian brain. And then this becomes this sort of green structures here, which includes things like the tectum, tegmentum, and the cerebral aqueduct, which is important for being able to process through that cerebral spinal fluid. We've talked before about that cerebral aqueduct. If it becomes blocked, that can lead to something like hydrocephalus, which is excessive cerebral spinal fluid in the brain. Here we can see the hindbrain here, and that structure is right here. And then in a human brain, that becomes the brainstem. When we look at the spinal cord in a vertebrate embryo, of course, that's split into the myencephalon and the spinal cord. And then here you can see in a human brain, that's the spinal cord, which includes also structures upwards like the medulla oblongata and the fourth ventricle here. And then we don't have this in the vertebrate brain, but in the mammalian brain, you see the metencephalon, which is the across brain, which turns into this dark blue, which is what I was alluding to, which is like the cerebellum, pons, and fourth ventricle. Now, we're going to walk through these structures in a human brain just so that you guys can kind of see how these structures are sort of sectioned out. And we're going to start with the cerebrum, but we're going to talk about each one of these. So we're going to go through cerebrum, diencephalon, midbrain, pons, medulla, and cerebellum, all nicely color-coded here, but we're going to start with this blue cerebrum, and we're going to talk about what that includes. So the cerebrum includes all of this sort of frontal lobe area, right? So we have the dorsolateral prefrontal cortex. This is really, really important for those real high-level functioning, executive functioning, memory, attention, being able to make us be able to have those really high-level processing. The ventrolateral prefrontal cortex area, also very important for things like memory and attention, but included in here, and we'll talk more about this later, is language. And so that includes a very specific language area for being able to comprehend and speak. Now we have the orbital frontal cortex here, which is important for things like decision making, emotion reward, and if you do something like damage this area right here, that can affect things like how aggressive you are. That can affect how likely you are to become addicted to certain things. So that can all be impacted by, for example, a damage. And then we have the motor cortex here, and we're going to get more into that in more detail, and that's really important for output from the cortex to the spinal cord to be able to move your muscles. And we're going to get into more specifically in that in just a minute. And then your central sulcus, of course, is going to separate your motor cortex with the somatosensory cortex. Now, the motor cortex is, which we usually refer to as like the M1, this right here is listed as area 4. This includes the precentral gyrus, and these axons are going to project to the spinal cord and brainstem. They're also going to synapse on motor neurons to allow for us to be able to move. If you have a person who has had a stroke, for example, and have damaged the motor area, you're going to see that person likely having some serious movement issues. They might have partial paralysis or issues with moving their limbs. Of course, we also have the premotor cortex area, which is before that, and then we have the supplementary motor cortex, which we will get to in the next slide, which sits essentially behind that motor cortex here. And this somatosensory area, very important for being able to understand and sense the environment around you. So, for example, it's really important for things like pain and temperature, being able to, have a sense of your environment around you, being able to, be able to experience your environment. Proprioception is this kind of idea of being able to have a sense of what's around you. And if you get this particular function damaged, that can really mess with your balance. It can mess with where you are in space. Think about when you close your eyes and you can kind of, feel things around you, and you can feel your balance. And it's harder to experience that without vision, for example. But being able to sense where you are in space is really important for you to be able to navigate your environment. Now, when we talk about the somatosensory cortex, okay, we often talk about how it correlates to where you feel sensation on the body. And so this right here, this kind of strain, there's an image called the homunculus. Essentially, it's a picture where you see a really large head and really big hands and tiny little feet. And what that is it's a demonstration of sensory input and how there are different larger areas of the cortex devoted to different parts of the body, and it's not an equal situation, right? So if you were to think about, let's look specifically at the somatosensory cortex. If you were to take a needle and poke the lips, okay, that's going to feel differently than if you poke, for example, the trunk of your body. So see, this is a very small representation here on the cortex. The lips contain a lot more neuronal connections to allow for a much more sensitive touch compared to other parts of the body. Some other areas that are very sensitive, of course, are the genitals in relation to kind of this idea of where they are. The tongue has a lot of connections. The thumb and hands, as opposed to, like, for example, the shoulder and arm, very different connections. And the same idea with the motor cortex and being able to think about your dexterity and how you're able to move. So like if you think about your hands, being able to grip a pencil and be able to write with your hands. This area of the cortex devoted in the motor cortex to the hand is very large in comparison to, for example, the toes. Now, let's say something happens and you lose an arm or lose a hand. Well, then this area of the cortex that's devoted to that particular function, that motor function, might be taken over by, let's say, for example, the toes and the feet. And so that's why you see people are able to compensate. This is kind of neuroplasticity at its finest. You see that the function can kind of take over one area that, if, for example, something's lost. And so if a person loses their hand or they lose some fingers, then if they're actually able to use their toes and use their feet to get around and to pick things up and to even write and play instruments. There's there's Olympic athletes who actually use their feet to use a bow and arrow. So this can definitely be something that can redistribute based off of certain kinds of injury. And here, this is just a sort of a picture of electrical stimulation. And if you have an open brain and you're able to stimulate different parts, you will either feel them depending on where they are associated with, or you can stimulate that. For example, move a hand, move a finger, depending on where you're at. Now, as we move back towards the back part of the brain, now we're talking about the visual cortex, and this is sort of all of this area is the occipital lobe. Now, we know that there are very specific projections from, for example, the retina through the lateral geniculate nucleus of the thalamus, which is located subcortically, and then that information is going to then be sent to the back of the brain to process. And we've talked about this before, about how important it is for the brain to be able to process this information in what we refer to as like the V1 area, to be able to translate it to a language that your brain is going to understand as visual information. And so if you damage your eyes and damage the input in, sometimes there's ways to go about to treat that to allow for stimulation and input of visual information, of light information into the system. However, if you damage the visual processing system, you can have a perfectly intact retina, eyeball, taking in information, but if this sort of translator is damaged, then you're not going to be able to identify what you're seeing. You're not going to be able to translate that information. So if you have damage at this level, that becomes really, really difficult to fix in terms of your occipital lobe. When we talk about audio, hearing sounds, okay, we have the superior temporal gyrus. So here, this would be like the temporal lobe. We have Wernicke's area here. We have Broca's area here. Both very important for language, being able to create language and comprehensive language. So being able to hold a conversation versus being able to understand language. So one area is really, really important for being able to understand language, and then another area is really important to be able to produce language. And often what we find is if a person has a stroke, they might have a particular kind of aphasia, which is a difficulty understanding or comprehending or creating language. And so that can be really problematic. And typically what we find is that it overlaps, and there are different types of aphasia. And a lot of times you can see that a person might have both difficulty comprehending and putting together sentences. It may or may not affect their ability to read. It may or may not affect their ability to write. But this area is really important for that information coming in through the ears and processing that into a language our brain understands. And then these areas are really important for language specifically. And what's really interesting is that this is really one of the few things that's really isolated to a particular hemisphere. So a lot of this these language areas are really confined to the left hemisphere. So if you get, for example, if you have a stroke on the left side of your brain, in these areas here, anywhere like right here, then there's a potential chance of having language difficulties. Whereas if you were to have a same stroke in the same area on the right side, the chance of it being language related is it's a lower risk. Of course, we have the olfactory system, and this is really important to be able to understand what you're smelling. And here you can see that this area here, which is basically a brain that has been flipped on its belly, if you will. These areas are really, really closely located to the nose. And these are the olfactory system is really, it's different in that it's the only system that actually projects directly to the cortex. And it is linked to the orbital frontal cortex as well as the limbic system. And if you think about the limbic system being really important for things like, being able to process emotion is really important for it, tied to memory, things like that. And we think about how important it is for us to be able to identify certain smells. We're really good at being able to smell certain smells, but not necessarily great at identifying what those are. But if we have, for example, like a toxic smell, we know that that might be dangerous. And so, or if we have a smell that we associate with somebody, that might be tied in to very happy feelings. And anytime we smell certain things, we might feel nostalgic. These are all because of these close ties into things like the limbic system. Now, this tie into the forebrain with the olfactory system, this is really the olfactory bulbs are really responsible for our sense of smell. And these structures, for example, like the amygdala, dorsomedial thalamus, these are really important structures, principal structures in the forebrain and the connection to the olfactory system. And if we look at how the size of our olfactory bulbs and the tract, it really is very small compared to other animals. And other animals like rats, cats, dogs, for example, really, really, really depend heavily on their sense of smell and are able to pick up scents that we are not able to. And so depending on, of course, the need to be able to recognize other animals, to be able to smell certain even like pheromones and things like that, we're just not as aware. It doesn't mean that we can't smell them. It's just that it doesn't meet our threshold enough for us to maybe be able to identify them or to identify that we're actually smelling something. And so these systems are definitely more evolved and larger in animals that are more dependent on the sense of smell. Here, of course, is the sensory cortex. It's you can see that it's kind of sectioned off based off of motor versus sensory, like somatosensory. And so the association cortex is all this kind of really lighter pink here. And we can see, for example, is involved in like olfactory. Here we have in the darker purple, we have the secondary sensory motor areas here, which are in this dark purple, like the pink, which is like the premotor areas here, the sort of posterior side of the somatosensory cortex here. And those are associated with of course, kind of the secondary factors there. And then here we have the primary motor cortex, which is in blue here. This is really important for motor. And then, of course, we have the somatosensory system, which is separated by this gyrus here, which is very important, or sulcus here, the central sulcus, separating those two. Now, if we look at the limbic system is a system that's important for emotion. It's important for memory. It has some of these really important structures that aren't really visible when you have the brain out and not, if it's just kind of sitting there. You're not going to be able to see these subcortical structures. So we have the cingulate cortex here. The corpus callosum right here is what connects the two hemispheres together, not included in this. We have, for example, the parahippocampal gyrus here. We have the thalamus right here. And these areas like the cingulate gyrus, the parahippocampal gyrus, the foreexternal gyrus, hippocampal formation, all important factors of the limbic system, as well as structures like the amygdala. The amygdala and hippocampus here, which are located, as you can see, we have the hippocampus right here and the amygdala sort of right here. Okay, the hippocampus is shaped like a seahorse, and the amygdala is kind of on the end. I've showed that in class with the app where we kind of looked at the left and right amygdala. Amygdala is very important for things like emotion. A lot of people just associate the amygdala with fear, but it's much more than that. It's not just fear. We know that some of the studies that have been done with the amygdala and fear components, they need to be replicated, and we know that it's much more emotion than just fear itself. We know that, for example, if we sever the amygdala in a cat, they before the ablation are likely scared of a dog, and scared, or the same thing with a rat. If you take a rat and essentially ablate both the bilateral amygdala, you're going to see that the rat is no longer scared of a cat scent. But prior to the ablation, they were scared. And so it's unclear if these are projections. It's not necessarily is fear housed in the amygdala. Probably not. It's probably to do with the limbic system overall. We know that the amygdala is heavily connected to the orbitofrontal cortex, which is important. The frontal area is really important for those higher level functions. And we know, of course, that the hippocampus that's involved in the limbic system is really important for learning and memory. And these systems all work together to allow us to be able to learn from the environment around us. If something causes us fear, we tend to remember that. If we have a fearful response, we tend to avoid that thing in the future. So these are all really important for our own survival. The hippocampus here, you can see kind of wraps around here, the hippocampus, and then the amygdala is on the end here. Really important for memory and being able to form long-term memories. Many of you are probably familiar with the case of HM, who had severe seizures, and the surgeon actually excised the hippocampus, and he lost the ability to form new memories. And that taught us a lot of information about memory and about how important this system is. And are your memories housed here? No, because HM was able to remember past memories, and he was able to formulate new procedural memories. So we know that procedural memory process must not necessarily involve a hippocampus. And so we know that, based off of these cases, that the hippocampus is important for creating memories, this process of creating memories. But, are memories stored in the hippocampus? Then I would say that is absolutely not the case. Here, the basal ganglia, we talked a little bit about this before. This is really important for the control of movement. If we look at some of the structures of the basal ganglia, okay, so for example, the caudate, putamen, globus pallidus, if there are injuries to these particular areas, we're going to see potentially disorders or problems in movement. One very specific area of the basal ganglia that we've already talked about that is involved with Parkinson's disease is the substantia nigra. And so Parkinson's disease, one of the primary symptoms of Parkinson's disease is the inability to be able to initiate movement. So an individual with Parkinson's disease has something that's a very commonly referred to as the Parkinson shuffle. And so they sort of walk with their feet. They don't really pick their feet up because once they lose the momentum, it's very difficult for people with Parkinson's to be able to continue to walk. Same thing with like brushing your teeth. You need to kind of start the movement, and we've learned that the basal ganglia is actually, specifically the substantia nigra, is really important for that. And so the substantia nigra, as you can see, that's located right here. And here, this is actually a slightly darker color. If you were to look at this in a, for example, if you have a person and you're looking at dissecting a brain, that coloration is actually caused by the dopaminergic neurons that are throughout that particular area. That's a specific pathway, and so that's really important for one of the specific dopamine pathways. And in a person that has Parkinson's disease, that those dopaminergic neurons are no longer working properly. And you can actually see that the coloration is lighter in a person with Parkinson's as compared to a neurotypical brain. We know that the basal ganglia is not necessarily controlling motor movement directly. It's involved in the process of motor function. And looking at, for example, the corticostriatal loop, very important for monitoring motor activity involved in the process. This is one of those things where you're looking at the brain and you say, well, does this area of the brain control this? And it's unclear sometimes if that's where the function is stemming from, or if there's a projection that, if severed, could impact that function. And so it's not necessarily fair to say, okay, this particular area of the brain controls this thing. It might have projections that are really important for function, and if those projections are damaged or injured or severed in any way, then that is going to be indicative of a particular symptom. So we have to be really careful with how we go about looking at this particular area is responsible for this, because if it's a modulating or monitoring type of situation, it might be a portion of the process versus causing that function directly. So I'm going to pick back up on the next section. I'm just going to kind of split these videos up a little bit just so that you're not having to watch so long. So we're going to